So after having excluded the mobilization, we came back to our kind of core model, and I used this model to predict key properties of the EPO receptor system. And I want to focus on two different points. So first, when we look here with this dark red curve at the concentration of free ligand in the medium, you see that there's a rapid depletion. So after 5 hours, there is no more ligand in the medium present, so the cells cannot be stimulated anymore. And on the other hand, free receptor at the plasma membrane, after a very steep initial drop, goes back to normal levels after 5 hours. So meaning first that there is never an absolute refractory state, so the cell is always responsive to ligand, and it gets back to normal levels very quickly. So if you now want to work with such a model, you first have to show that it's really valid to describe your data, and therefore you make predictions and then validate them going back to the lab and doing experiments. So we first wanted to validate that EPO indeed is rapidly depleted from the extracellular medium. So we therefore performed immunosuppressive mutations. So we enriched the ligand from the medium and looked at the kinetics over time. And you can indeed see that we have this predicted rapid depletion of ligand in the medium. And we cannot only show this for the murine EPO receptor, which is our standard model system, but we could also show that it's true for the human EPO receptor. And furthermore, we could also confirm these results in primary erythroid progenitor cells that you can isolate from mouse embryos. So this ligand depletion is a very central characteristic of the EPO receptor system, and you can find it in both in the murine and human system, and also in primary cells. And it already shows you that the signal initiation, so at the very beginning of stimulation, is regulated by EPO receptor endocytosis through ligand depletion. So this is the main action of receptor ligand internalization. So we also took another approach to validate these model predictions, because if you have such a situation here, that means that the biological activity in the medium is decreased over time. So what we did is we performed a time course with cells and enriched for the receptor by immunoprecipitation, and then looked at the receptor phosphorylation status. And you can see here a typical time course experiment in this first half of this immunoblot, showing that there is an initial peak of receptor phosphorylation, and then it drops back to basal levels. Now, when we prepared the immunoprecipitations from these cells, at the same time we took the medium and used this to stimulate fresh cells. We stimulated them all for 10 minutes. So they all had the same chance to get phosphorylated at the receptor level. But what you can see here in the second half of the plot is that the later the medium was collected, the lower the phosphorylation signal for the receptor gets. So also confirming that the ligand is depleted from the medium and that the biological activity is down. So knowing this, we also could actually validate that there also is an EPO receptor recovery at the plasma membrane. So we again performed a time course experiment and looked at phosphorylation levels of the receptor. And in blue, we again have just a very normal time course experiment. And for the red samples, we added ligand just 10 minutes before we took the cells for lysis and immunoprecipitation. And the hypothesis is if there is receptor at the plasma membrane present, you should see this if you add new ligand, if you give the chance for a new stimulation. And this is exactly what you see. So, for example, here 300 minutes. In the normal time course, the levels were back at basal levels. However, when you add ligands just 10 minutes before, the receptors were phosphorylated again. So, showing that the cells always remain ligand responsive. Now, after we could validate our model with these experiments, we used it to perform model simulations and to actually look how the system responds to very different ligand concentrations. And therefore, we plotted such a curve for EPO receptor complexes at the plasma membrane over time. And we did this for many different EPO doses. And then we could derive two different quantities from these plots. So first, we could look at the peak amplitude, so the highest point in receptor phosphorylation, and we could also calculate the integrated EPO receptor occupancy, so the integrated amount of receptor ligand complexes over time. And when you plot the amplitude, you can see that you have a saturation curve. So for higher ligand concentrations, you do not get more receptor ligand complexes at a defined time. However, if you plot the integral, you can see that there is a strict linear relation between the integrated amount of complexes at the surface and the amount of ligand. And this linear response curve goes beyond the point of saturation at the peak amplitude. So this already indicates that the EPO receptor system indeed reacts to ligand stimulus in a linear fashion and not in a saturation-like kinetics. So now linking these findings to experiments, we performed time courses and looked for phosphorylation of the receptor and phosphorylation of the associated kinase JAK2. And again, we did this for different EPO doses and derived the peak amplitude and the integrated amount of phosphorylated protein. And you can see that the results perfectly mirror what you got for the model simulations. So meaning you have a saturation for the amplitude. However, surprisingly, you also have a linear response curve for the integrated amount. So this linear relation is not only valid for the complex formation, but also for the phosphorylation stages at signal initiation at the receptor and kinase level. So coming back to our four hypotheses we had at the beginning, so we could rule out the first two, the abundance and the mobilization. Now we are left with the two last hypotheses, the recycling and the turnover, as means to achieve a linear response curve. So to answer the question which of these two hypotheses is utilized for the EPO receptor, we performed model simulations and started with the recycling. So when we plot the integrated amount of receptor ligand complexes at the plasma membrane versus their amount of ligand in the medium, you can see that increasing or decreasing the recycling rate does not change the linear relation between these two quantities. Just the slope is changing. So actually recycling is not responsible for the linear relation.